I was asked today to actually talk about yourself, which normally actually you don't actually do that often. So the first part of the talk is a rather circuitous route that I've done medical research. So I haven't followed a traditional route. And then secondly, a talk about the trajectory of medical research, how you start, continue, and hopefully survive. So there are two separate components. But firstly, what's a clinician-scientist? Well, I think the first thing is you have to be good as a clinician. So you have to have rigorous clinical training and practice. And you have to be good as a scientist. Right? So it's not a soft option. And so one of the things that you bring as a clinician is that your research is informed by your clinical experience. And so a lot of the research, which I'll show you now, has been done by scientists who I work with very closely. And sometimes you think, what are you bringing to the table? You're bringing to the table a clinical perspective. But for the scientist, you actually need rigorous scientific training in research methods, whether they're laboratory, clinical, public health. And I actually think realistically, you need a time when you step away from clinical practice to immerse in research training and to learn that, and then to have ongoing research. So I started out clinical training here. I kind of was born in this hospital, worked in this hospital, left it on 3 occasions, and they somehow or other re-employed me. My wife thinks the inevitable will happen and I'll die in this hospital, which is probably the case. So I was a resident here, and my wife is a geriatrician, is also a physician. We went to Port Moresby, and I've always been interested in global health. I did a year of general pediatrics, and I have to admit, I thought, well, that'll do me for Nepal, which also I apologise. I then did tropical medicine in Liverpool and learned about worms and bugs, and did an MRCP. And then I went to a rural missionary hospital in Nepal, in Western Nepal, for 3 years. And this is a hospital built on the side of a hill. It was a 100-bed hospital, and we had a population of about 1.5 million people who were served by the hospital. We had a surgeon, fortunately, and my wife did the obstetrics, except when she was having a baby there, who was said Philip Britton, and did general medicine and pediatrics. And in the monsoon, it was very fine. And actually, this was the view from our house. I could tell you about many of the things that went wrong, but we won't go into that today. So I was there for 3 years, and then I wanted to, well, we were overwhelmed by tuberculosis. We would have about 1,500 TB patients and 250 leprosy patients a year. And I nearly ended up going and working with very famous clinical trials called Wallace Fox in the British MRC. But in the end, I came back to Australia. I actually started working in respiratory epidemiology, did an epidemiological study in childhood asthma with Anne Woolcock, but I was really not a public health person. But this is leprosy. In the black and white old photos, what's called a type 1 reaction in leprosy. Leprosy is actually an immunologically mediated response to a very weak pathogen. And on the other side is a different type of reaction, completely different immunological reaction called a erythema nephrosim leprosorum. But I came back to Sydney. I realised I wasn't a public health, so I started doing the wrong PhD, but Anne kept telling me to be all right. And we parted friends. And then I wanted to work on leprosy. And of course, you know, that was in 1981, 82, thought to be a pretty silly thing to do. But I had this clinical interest. Now, you probably don't know that the leprosy bacillus, which was the first bacterium associated with the human disease in 1873, can't be grown. We still can't grow this bacterium. because it's so adapted to live in macrophages and Schwann cells. However, and this was the case, however, immunological tools had come along so that we could actually study the organism without actually growing it, which still is surprising. So I had to find a supportive mentor, and fortunately, Professor Baston agreed for me to do this project. And then I had to have some scientists who would teach this rather kind of jungly doctor from Nepal how to do basic cellular immunology. And this photo, which was this cartoon, which was drawn by Chris Goodnow, a very famous immunologist, is of an armadillo. And I was able to do a PhD because M. lepre grows in armadillos. That's another fact for the day. And it had just been discovered how to purify M. lepre from armadillos. So I wrote a begging letter to WHO asking them to give us some M. lepre. And then I collaborated in what was called the Circus, the Clinical Immunology Research Centre at the University of Sydney. And we all had a place, and many clowns, etc. And over the next 4 years, I made a whole bunch of monoclonal antibodies and discovered 6 antigens, which kind of could do because there were only 2 other labs in the world doing this. And that was very rewarding time, and where you learned, as I think Cheryl said, really basic science properly. And I shared these antibodies with 9 different labs around the world. We didn't have MTAs in those days. You just sent off the antibody and see what happened. And we isolated the first gene for a heat shock protein and the first M. lepre specific gene. And eventually, we isolated these genes individually, but the whole genome was sequenced in 2001. So at the end of this, instead of going to Boston, I went back to Nepal and went to a leprosy mission hospital outside Kathmandu and set up a research lab and learned how to kind of import gamma counters, radioisotopes, and do basic cellular immunology in a lab. And this laboratory continues to this day. And since then, I've had long-standing collaborations. This is actually the lab. It's the old women's ward. And subsequently, when I've gone back there, I've always thought how grotty it is. But we were able to do really terrific science. It has actually been destroyed by an earthquake now, and we're rebuilding another lab. So if anyone's got a spare $100,000 today, please give it to me at the end of the day. So we established this lab. We evaluated recombinant antigens as diagnostic tests. We did seroepidemiology. There's a very ancient assay to culture M. lepre in mice that we did. And we studied the immunological mechanisms of leprosy. And as I said, this continues today. And because we'd set up this lab in a place where there was lots of leprosy, we collaborated. This is a study actually done with colleagues from the Rockefeller Institute looking at the effects of recombinant IL-2 in leprosy patients when I was a bit younger. Okay, so then after that, by this stage, we had 4 children, and we returned to Sydney, and by this stage, the circus had become the Centenary Institute. And fortunately, and as Cheryl emphasised, the importance of a mentor who provided a venue and support for research. So I established another mycobacterial research group, and we've gone on to do a lot of research. We continued leprosy research until people would stop funding it because they didn't think it was a big deal, and we continued working in Nepal. But my interest was always this condition. And these are all examples of TB prior to HIV in Nepal, in children, causing up extrapulmonary TB, severe malnutrition, and severe disease. And so a couple of points about, I guess, the middle stage of a research career. It's actually the importance of adaptation, a bit like the immune system. you have to be prepared to adapt. So we moved into TB research, and in the Centenary Institute, we had people making knockout mice. So we characterised the role of TNF and TNF superfamily members in the control of TB and granulomas. And of course, that's extremely important in the use of biologics. We did some immunogenetics, and we particularly were involved in vaccine development, first for leprosy and then TB, and more recently COVID. And also developed other collaborations. So I'm a believer that in medical research really comes from scientific research at a laboratory level. So I've been very fortunate to collaborate with very good chemists and been involved in structure-based and natural product-based drug discovery. We've made synthetic vaccines, and with Kim Chan, we've looked at pulmonary delivery of vaccines. And to do this, you have to actually have a containment facility where you can work on TB. And so I've been involved in designing 4 different PC3 laboratories, including a new one in the SBA building. And we recently have got a contract from NIH to look at doing head-to-head comparisons with 9 different vaccines over the next 5 years if we keep going. So firstly, it's very important to adapt. Secondly, it's very important to collaborate. And so with Guy Marks and Greg Fox, we've worked not with actually this person. This is a bust of Robert Koch, who discovered the TB bacillus 9 years after the leprosy bacillus was discovered in Vietnam in the National Lung Hospital. And we've been involved in a whole series of studies about finding active TB patients in the community. We call these ACT. So we're up to the 5th ACT, and we hope this is not a tragedy, and it keeps going. But these have led to very robust clinical trials which have proven the value of active case finding in the community. And we're applying this at the moment, actually in Kiribati in the Pacific, which has a very high rate of TB. So over the last decade, I've led 2 centres of research excellence in TB research, and this was the productivity of one of these. And these have been very much about collaboration. So both leprosy and TB involve all aspects of medical research, from sociological research, understanding stigma, understanding why people present with disease, right through to basic biological research. We've been involved in big collaborations in China with Carl Feng, and I have to admit our last collaborative meeting was in November 2019 in Wuhan, when we could talk about that at a later date. And these have been very productive with colleagues in China. And then, of course, COVID came along, and we had a PC3 lab. So with a lot of effort, we moved it to doing SARS-CoV-2 research, and we have the only animal model for SARS research in New South Wales, and have written many grant applications. And you have to get used. One of the things to be involved in medical research, you have to get used to failure. And so I lost count of the number, probably about 12 different research grants we wrote about COVID, but we got 4 or 5 of them and we've developed a nose-only vaccine, which hopefully will block the transmission. My colleague, Jamie Trickus, who originally started in leprosy research, is now leading this amazing international collaboration for a globally accessible COVID vaccine. And we've shown that at least 3 of the vaccines that Jamie has developed are protective against COVID. And with NHMRC funding, we're looking at the interaction of COVID and TB and chronic lung disease. And as said, I'm research director for the hospital, so I've been involved in RPAH virtual, and we've looked at the role of remote monitoring for COVID patients and been involved in a New South Wales collaboration. And this is just to show that this this one slide probably took the best part of 2 years' work to get done, showing that this mucosal vaccine for COVID is completely protective. Very interesting, actually, that the technology we use for this vaccine actually came out of a TB vaccine. Showing the kind of interaction of these things. So, and that's where we are now, working on COVID and TB in a number of different countries. So the second part, though, was to talk about the trajectory of research. And I guess I've been very fortunate to combine my interest in global health also with a kind of academic research in Australia. And that was really only possible through a couple of key people who supported you at a time, and then you had to prove the value of what you were doing. But the importance of having mentors who support you is critical. So I've divided this into 3 stages of a research career, and this is particularly in medicine. There are 7 stages, of course, in Shakespeare's play, As You Like It. I'm not quite sure which stage I'm in yet. So certainly, you don't want to think about the 6th and 7th stage. You just keep plodding along in the 5th stage. Okay, so there's a time for research training, research consolidation, and continuing in research. So I think, as Cheryl and Elizabeth were saying, if you're interested in research, you really should put your toe in the water. Before training, I'd done a BSc Med. I'd spent a year doing lab work and really didn't want to do it again. And that's why I took actually, whatever it was, 8 or 9 years to discover what I wanted to do a PhD. And that's probably not the most efficient way to do it. And you certainly need to have a long-suffering partner to do that, and someone who can support you while you're doing your PhD. But it is important to use every opportunity, and there are many opportunities for projects. There are many opportunities during residency or training and doing case reports, systematic reviews. It's good to participate in ongoing projects, understanding the ethics process, and research is really the ultimate team game. So understanding how you can have a place and can contribute to a team, and writing a manuscript, writing an HREC application. You never know where these things go. When we were in New Guinea, we found that unfortunately a way people could commit suicide was taking an overdose of chloroquine. So with a colleague, we wrote up 20 of people who'd had overdoses of chloroquine and what were the characteristics and this paper languished. No one ever referenced it. And then COVID came along. And there was this little phase of treating COVID with chloroquine. And for some reason, I was looking up something, and suddenly this paper had 20 citations from the 1970s. So you never know, just never know. Then starting out in research is you do need training. So at the hospital here at Prince Alfred, as research director, I set up a clinical research centre, and we have training in research methods and particularly data management. So, research, whether you're doing a clinical trial or whether you're doing qualitative research on why people take a particular treatment, it may be qualitative, but you have to think about it quantitatively. So you have to start to think about using data. You need to become statistically literate. And there are lots of people. You can speak with your supervisor, colleagues for defined projects. And we've found this clinical research centre to be used enormously. It was interestingly when we were very fortunate to employ a very good data manager who pointed out that the wrong way to do research, this is the one warning today, the wrong way to do research is to have a list of patients on a USB stick that you carry around in your pocket, right? in that you need to be able to have clinical data under appropriate controls. So we set up REDCap and have over 5,000 users of REDCap in the Sydney Local Health District. And, you know, the trial that we're doing in Kiribati is collecting data on patients with TB in REDCap. And we have a whole bunch of courses. So after you put your toe in the water, you need to become competent by requiring some full immersion. So there's a good example of swimming, Cheryl. And it could be in a range of things. And it could be done in a range of ways. I think there are changes and complexities in life now that probably it used to be to get a job in this hospital, you'd need a PhD. And that's a bit more complex. And there are, you know, masters, there are skill-based masters, there are research-based masters, and they can be used very effectively. And a number of our colleagues in many different disciplines are doing that. But it does, and the best way to learn to do research is to do research. So you actually learn to do it as doing it personally. And it requires planning ahead. And I following on from Cheryl, that your active members of the department, trainees, the clinical school, the research centre in your particular place, the places, there are medical research institutes such as the Centenary Institute, which is just behind us, where you can go and talk to people, and finding financial support in a whole variety of ways. And the one thing you keep doing in research is applying for money. So learning to do it at an early age is good. It really helps to be in a stimulating environment. And the best way to know what are the stimulating environments is to speak to your peers and colleagues, not necessarily your supervisor. You talk to your colleagues and you'll learn there are good places to work at, places with different potential. And the critical thing really is to be inquisitive, actually, to be inquisitive and to ask questions. And these are just a couple of comments from early career researchers at PA. I won't go through it completely, but it just makes the point that this person was afraid to dive in the deep end, but actually found they were warmly welcomed in the laboratory they went to. It's good, you know, to learn humility and to actually learn. You might have got on the top of your particular ology, and then you have to go back and work in the lab and relate and suddenly realize you don't know the right end of the PET, the right end. How do you use a SAS program to associate it to a clinical trial? But you'll find that you'll be welcomed. In fact, actually doing a PhD training is I think one of the most rewarding things you can actually do. Transitioning can be more challenging, as this person found. And we've been looking at ways in this institution of supporting that. So consolidating as an early career researcher, again, it requires planning as how your research training, and there is no right way. The way I did it's probably the wrong way, but it's the right way for you. And there's certainly changes in the way NHMRC and the Medical Research Fund research now, and knowing how, and research fellowships, knowing how those work is very important. And practically speaking, you need to require a portfolio actually, and I'm sure many are doing that. And as you expand your skills and network for future career, that actually expands the opportunity. And this is another person from here. Postdoctoral fellowship provides the space and relationships to grow independence, depth, and collaborative networks. And as Cheryl said, moving out of a comfort zone into a highly competitive North American environment can be extremely helpful. I was fortunate after we came back to Australia to do 3 different sabbaticals actually over the last 30 years in different places with workers, and they've been extremely helpful and would always encourage that. And in fact, for clinician researchers, this is a bit of a bottom line. It actually means turning down the apparent security of full-time clinical work. at a stage in your career. And I know this is complex, having 4 children who live in Sydney and knowing all the issues in Sydney, but it is the reality that if you make that commitment, the long-term benefits to your career are of great value. So what happens in the midterm research? I'm not quite sure if this is the right image to show, but there is attrition of clinical researchers and clinical scientists, which is widely recognised. So there can be this investment and then people can't use it. There are many different reasons. We won't talk about this. And there have been many thoughts. These were a series of papers in just before COVID, which were looking at how to address this around the world. I think critically sustaining a research career depends on adaptation and it depends on collaboration, adaptation and collaboration, and support at different stages of the career within your own department. And there are now actually many more resources around to think about this through AHRTCs such as Sydney Health Partners and MRIs. But it is important to build research collaborations nationally, internationally. So, and this is a clinical researcher at Prince Alfred. I feel that mentorship and being part of a research team is the most important aspect of establishing and continuing a research career, no matter what level you're at. And so to address this here, and Cheryl knows about this, we've decided within the clinical setting, and here I'm, I guess, specifically talking about physician trainees, that we've established an institute for academic medicine to promote research training of research clinicians and the professional development and support of researchers at all stages of their career. So finally, why do I work on TB? Well, it is the commonest cause of death from any bacterial infection in the world. Over 10 million cases, and COVID's put back the control of TB 10 years. So as this Nepali sign says, TB anywhere is everywhere. So that's why we work on TB. Thank you. Well, I guess our experience has been that we really do have to work and understand the culture seriously. And obviously we lived in Nepal for 7 years. So that's, but interestingly, actually, the research opportunities have come in Vietnam, China, and the Pacific, not so much in Nepal. So, and you need to actually work within national programs. One of the value of our research studies in Vietnam is that they're part of the national program. And we have a new program called Pearl in the Pacific, because there's this island nation which has one of the highest rates of TB and the highest rate of leprosy in the world. So that's called the Pathway to the Elimination of Antimicrobial and Latent Tuberculosis. It's very important to have a good name for your project. And we're doing that within the Ministry of Health and Medical Sciences, and our staff are actually employed through the ministry. So therefore, what you're doing is not just doing a research project to do some papers, you're doing a research project which, as well as answering questions which will have global significance and maybe setting up a model which is scalable and useful, you're actually leaving behind a kind of legacy within the national program. So that's a hard thing to do, to start out as a researcher, but there are many programs around the place. And going and joining those programs is critical. Always looking for willing workers.
Hi, thank you so much for the talk. I was wondering, so you mentioned that you were very passionate about leprosy, and then
Yeah.
unfortunately funding dried up. How do you balance the tension between doing what interests you and what you're passionate about
Yeah.
with what will be funded?
Yes, that's actually a very helpful question. Well, you have to be realistic, but I mean, I've chaired a research committee on leprosy for the leprosy mission for 25 years, and I've, spend my some of my holidays going and working involved with those leprosy facilities. And you've changed. I'm on the board of the leprosy mission, involved in the governance. It's the largest NGO working in leprosy. So you do take on different roles. And again, I think that's for a clinician researcher, when you take on those roles at a later stage, you actually bring both the clinical aspect and the research aspect to the governance of the organisation. And the research aspect is not necessarily, pushing your own hobby horse. It's more about thinking about innovation and change in the organisation, whether it's a big hospital like this, that it has to innovate and change, or whether it's how you run wound care clinics in a rural area of India. Both of those things, probably the latter is harder than running this hospital, but thinking about that innovatively with a sense of change is something that you can bring. So I know that sounds obviously to many people starting out in your careers, but I guess I'm highlighting that those experiences are never lost, because when you're at a later stage of your career, you can bring those things to the table.
Thanks for the helpful tips. Could you say a bit more about the Institute of Academic Medicine and how we might engage with it?
Yes, I think what I guess what I've observed is it is hard for full-time clinicians to sometimes use all of the resources around them. Even, we're right next to the university, but yeah, it's hard to go for a talk at 3 o'clock in the afternoon when you're doing a ward round. And so you need to develop structures within the organisation that recognise the importance of, in this case, it's not just about research, it's about more professional development and the utilisation of people's skills. So as a physician, I'm always reluctant to learn from surgeons, but it is the case that the Institute of Academic Surgery, which was set up here about 10 years ago, has been transformative in surgical careers. And that's an example where, for example, people doing a master's within a surgical program, there's no point in being a surgical researcher unless you're a good surgeon and a good researcher. Right? So surgeons are a bit different, a different species, really. But by showing that you can use master's training and bring actually innovation. One of the things surgeons are very good at is innovating. They're great widget users. They're really good innovators. But putting that into a kind of research framework where the innovation or the widget is actually formally assessed and the cost benefit is assessed and the effect on the hospital is assessed. So, we have seen that, and in a way which engages people who want to go down that direction from an early stage. So, in this particular place, we've found doing that. There's a similar one now in women, children's and families, and a similar one in precision medicine. So it's really actually bringing the opportunities very close to where the clinicians are working. And so that's actually, yeah. Yes, yep. I mean, we started this all of 2 months ago. We were going to start it in 2019, actually November 2019, but something's intervened for the last 3 years. So, but we're starting it now. Yes, and that is very much an opportunity. One of the things and I'm sure wherever you work in large students in large organisations, it's really sometimes difficult to crack into the organisation, you know, and some people are extreme extroverts and do it. Other people, you know, just find it hard to go up after grand rounds and say, listen, you know, can I work with you or something? So it's actually it's more about, you know, creating opportunities for people to get the benefits out of the organisation where they're working. That's the kind of. Yes.
Thanks, Professor Britton. I wanted to ask if you could speak a little bit more on the transition from early career research to mid-career research and what makes it so challenging.
So I think it's often said, actually, it's hard enough to get your first NHMRC grant. The hardest one to get's the second. The reason is, you know, you often are on a high. I always think about it that the PhD student doing a project should be right at the front of the wave, a particular project. And you should know more about your project than your supervisor. And you should feel a bit, you know, what's this old fuddy-duddy doing? They don't really understand what's the critical issue. Now, some students get there, and they're the ones who are now professors all around the world. Not everybody gets there, but in other words, but you really should be at a bit of a high. And then that momentum takes you to a postdoc or gets you a new job in a different hospital. You might get a new grant, and then the realities of clinical work, administrative work, you know, filling out forms 3 times in triplicate and all that stuff. And you have to learn to find ways to do that, but actually also find time to drive your research. And so I think, and that is where working in a department or a team or a group of people who recognise the importance of bringing that young person on is important. And in a big place like this, you can see departments that have been very good at doing that for the last 50 years. And then there are some departments, I have to admit, there is 1 or 2 departments here that could improve. And so, and there are many, many reasons for that. But so I think at that stage, that it's not almost mentorship, it's also people creating the space for you to be successful in that new role. And that success then leads to the second grant.
Hi, we got a question from online. How did you manage having family and working overseas?
Well, 2 of our children were born in Nepal, and so that takes a bit of long suffering, and 2 of them were born here in this hospital. I guess because we wanted to do it. Yep. I mean, you know, times are different a bit, but I guess children have just done what we were doing, and well, in fact, it was all part of a family activity. You can probably talk to the children, and they.
Warwick, I'm sure you had a major role in
Yeah, I mean, it is a partner. I could not have done nothing like this if it hadn't been for Annette. But she's a very good clinician. She's very involved in resident training, was the director of clinical training at Canterbury and then at this hospital for 10 years. And so I guess we've had complementary roles. I think, yeah, children are part of the family, not a entity unto themselves. I think if you're doing things together as a family, it takes along. The thing, we've always had 3 weeks holiday a year. 3 weeks holiday a year. And that actually, even in odd places, trekking in the mountains or travel a long time on trains in India, I think that is actually very important Yeah. Yeah.
Well, I also think what is important is realise that here or there.
I think the other thing, certainly in my own laboratory, I've had people, current postdoc who's taken a year off to have a child, and one creates the opportunity for her to come back part-time. And my experience is that, as I'm sure you all know here, that women who work part-time are some of the most efficient people in the organisation, actually. And that would be true of my own wife as well. So I think, again, it's part of the team, and talking about these things. People sometimes would feel a bit reluctant to talk about it. Can't necessarily solve all problems, but sometimes you can have very practical solutions. Personally, my particular bugbear is I think the colleges have taken too long in the way they've lengthened their training. It doesn't take you 5 years to learn how to do gastroscopies. I could do it in 6 months, really. And I think so. It's being and we left before we completed our FRACPs, which was thought to be a total failure, but we were managed to fill up the time and fix it up later on. So I think looking at ways where you can intercalate things is also very helpful. And personally, I actually think the academy should really be lobbying the college to have a much more flexible approach to research training. That's yeah.
It's happening slowly.
Yes, yeah.
Just one last question. Just in terms of those career breaks, especially in shifting between career research.
Yes.
A lot of the grant schemes, they do have like early career, like within so many years of submitting a PhD. Like, do you have any advice for people who do have a career break, or they're working part-time, part-time research, but then they still need to be competitive enough to get a grant?
Yes. Yeah, I think it comes back, and again, I'm no oracle on these matters, I can assure you. But I think part of it is not seeing yourself as an individual, but being part of a team that supports that. So sometimes it is the case that some researchers may actually take a more support role than others. Others may, and within a program, they may contribute to a specific area. I think actually the NHMRC is recognising people taking time out much more adequately than in the past, and I think, and it's a way to actually stay involved. For example, just thinking about this in my own lab at the moment, with one of our really good ECRs who's on leave at the moment. We have a scheme where they can come in for 10 days during that time and spend a day working on a paper, or spend a day coming to a meeting, and maintain a focus. So I think there are different ways that you can do it, but it's difficult to do by yourself. It really need to be part of it.
Sure, last comment. There's no one right way to do this, and I think it depends on. So firstly, I agree with. So I agree I agree with Warwick that there is better recognition of career disruption for any reason out there. But I think it's also thinking strategically about the type of research you're doing and how to keep using that time well to be productive throughout that period, because in reality life gets in the way. There's no one straight path, and it is tricky, you know, when it's almost easy to get those first grants, you know, in those early periods of time post your PhD. But the next period, it's really thinking about where do I want to be in 5 years? You know, what is the research I want to do? What is the career I do? How do I do that? And it might be that your path is different, you know, to someone. It might be that you're not the next MRC investigator grant, but really, you know, if you can be doing the sort of research you want to do in a group you want to be in, you know, it might be that you'll be there further down the track. But in this period, while you're managing all these other things and making the most of that time, you know, that might not be the path for you, and that's okay. You know, Warwick said, and his isn't a career to follow, and you know, if I gave you a long history, mine's probably not either. But there's no one right path, and there's no time frame to get there. I think that if you can be thinking, what do I want to do next? What's important? And trying to be as creative and getting as much advice from different people, you'll get there.
Thanks, Cheryl. Could you carry in thanking Warwick for a fabulous session?